So the first section of our do's and don'ts is going to be our abstract to our paper. Now, before I get into some tips and some things the abstract must contain, can anybody tell me what do you think should be in an abstract based on what you know? Yeah.
Well, an abstract should basically be a summary of your whole research paper, so a person who's reading it can get an idea of the results from it. The whole paper, so it's just a summary of everything that occurred in the paper.
Awesome job. Have a candy.
Oh, that was a
I can't throw.
horrible-
I warned you.
I'm
Okay.
on that. I'm on that.
Anything else before we get into it? Anybody else have anything that they think? Okay. Well, an abstract, as you mentioned, is the first thing that your either reviewer, if you're writing for publication, or your professor is going to read. Okay, and the key to these things is they're short. Okay, you don't want this to be more than 250 words. That's for in terms of maximum length. Minimum length, I've seen abstracts that are a couple of sentences long. But you want to make sure that they convey, as you mentioned, what you did, so what was done in your experiment, how you did it, and what did the results tell you. And these are going to be about a sentence or two apiece. So you want to make sure that it contains that and not too much longer than that. And remember, you want to sell your paper here, okay? This is the first thing people are going to read. They have to want to keep reading. That's what's going to make them read your paper, and if it's a professor, it's going to make them interested and get you off to a good start in terms of grading. So it means to avoid in your abstract. You want to avoid a lot of copious backstory. You don't want to add background and stuff that belongs in the introduction in this section. You don't want to add figures either and pictures. This is a text-only section of your report. So no tables, no graphs in the abstract. This should be, simply as I said, it should be only text. No citations either. You don't want to cite anything in your abstract. You're going to have plenty of time to cite things in your introduction, your discussion, and your method sections. So some helpful tips that I find when I write my abstracts personally, I like to write them last. You don't have to. I know people who don't, and it works perfectly fine. I write it last just simply because I think it's a good way. You've already written your introduction out for your background. You've written your materials and methods for how you did it, and you've already written your results and discussion for what did you find out. So that's why I like to put it in last to tie everything together. And the other thing, the big thing that I find is have somebody else read it. Okay, I've had my peers read my abstracts and they look at me and they're like, this is not what it's supposed to be. And it made perfect sense to me, but when you give it to somebody else, it doesn't always convey. Because you're doing the experiment, it's going to make sense to you. And just because it makes sense to you doesn't mean it will to everyone else. Okay, so that's the abstract in a nutshell. And then introduction is next. So before we get on to talking about it, who can tell me what do they think belongs in introduction? What should the introduction have? Anybody?
More candy.
Yeah,
There's candy on the
free
line
candy,
here.
guys. No? Okay. Well, basically, you want to talk about what the problem is and what the background to your project is. So this is where you're going to convey that background. You want to tell your readers, why you're doing this. What's kind of been done already on your topic. And you want to answer the following questions. Why is this important? What about this experiment is important? Why did you do it? What was the point? You want to answer those questions here. And I say this is audience dependent. And the reason I say that is because there's different kind of degrees to scientific writing. You can be writing for very specialized fields of people that are very familiar with the type of experiments and the type of work that you'll be doing. And here you won't need as much background because they'll already be familiar with it. But if you're writing for more general journals or a more general audience, you're going to want to have more background so they can be caught up on some of the information that's already out there relating to your topic of interest. So that's why I said more general audience equals more background needed. Does that make sense to everybody? So citations. This is where you're going to find the bulk of citations, or at least a bulk of citations in your paper. You're going to everything that you add in this section is presumably, or most things, are going to be from other primary literature sources. So you're going to have to cite those. Okay, you're going to have to tell people where that you got this information from, so that they might be able to go back and look at it for themselves. So some helpful tips, provide some statistics in your introduction. Okay, if you're writing a paper on, some type of cancer cell line or a protein, variant in cancer, describe some statistics and how common that cancer is. What's the mortality rate? How many people get it per year? That type of information. Just make sure that you're using reputable sources for that information, other primary literature, not Wikipedia, never. So that's why, add some figures as well, add a graph maybe of some work that has already been done previously on this topic. If you're working with flies, all of you will have to do the fly lab when you're sophomores. Throw in some pictures of flies. Show what traits you're looking for in your introduction. So this is the spot where you can add some pictures. Just make sure you cite everything. And then again, that's optional. You don't have to do it. I personally like to, just because I feel it cements kind of the talking points in this section of the paper. And again, cite everything. Any questions on either the abstract or the introduction before I hand the floor over?
All right. So now we're going to get into methods, which this is kind of our nitty-gritty technical side of our report, right? What kind of things do you think we should include in methods? Any ideas? Shay? Right. Oh, okay.
Okay,
Yeah,
okay.
exactly. Steps. You had your hand up too? Right. So exactly.
Do you want
we
her
want to
candy?
put
You want her candy? Okay.
So we are going to want to talk about how did we solve the problem. We started by stating, what is our problem in the introduction, and now we're going to talk about how did we solve it. So with methods, we're going to talk about all of the steps of our experiment, right? But we don't want to list it out as if it were steps or a recipe or even just a list. So we don't want to have anything like that. We want what's called a narrative of our procedure. So what do I mean by narrative? Anybody have any ideas? Third person? No, not quite. I can see where you're getting that from though. No? A narrative just means that it's a continuous stream of thought, that we're showing why these things are related to one another. So based on the end goal of solving our problem, we want to show why these steps that we're going through are relevant. Why did we isolate DNA? Why is that important to our experiment? And you want to be concise about what you did in each step, but you also don't want to make it into a recipe. So although giving measurements is a good, accurate thing to put into the report, you don't necessarily need to say, I put this much in a beaker. We know that you had to have measured that into some kind of container, right? So be precise, but don't go overboard. So before, young man in the back mentioned the third person, or what's called the passive voice. This is something that a lot of you guys are familiar with when it comes to lab reports because that's what we deal with in chemistry. Absolutely, in chemistry department under ACS form, you should be using passive third person voice. But in biology, it is more appropriate in context to use active voice. So here I give the example of a phrase. In passive voice, we would say DNA was isolated from the culture. In active voice, we would say we isolated DNA from the culture. Now, everybody's probably like, whoa, wait, you just used we in a sentence for lab reports. Although, generally you're taught not to use I or we at all, you should use it occasionally when appropriate. It should not be constantly present. You should restrict your use of I and we, but where it's appropriate, you can say, we followed this procedure, or our experiment, our results. These occasional phrases are acceptable and help put your methods section into an active voice. So just like we talked about audience evaluation with the introduction, we also need to consider our audience when it comes to our methods section. For example, if we're writing our fly paper, if we're writing this paper to be directed towards other scientists who work with flies, we wouldn't necessarily have to talk about in detail the care and maintenance of fruit flies because our audience would already have this information. So we don't need to provide that and take up room in our report with such information. But that's generally at the discretion of your professor. If there's any question as to who your audience is and how much information you should be including, talk to your professor because depending on the assignment that could change. And additionally, using strong scientific vocabulary is really what's going to help our methods section. It's going to make our methods sound more professional and concise, and it's also going to cut down on wordiness. Using our everyday terms that we would tends to make a sentence much longer than it needs to be. So some useful terms that I've included are words like aliquot, to aliquot, a verb. It's a similar term as to measure. You generally use this verb for measuring the measurement of liquids. Like I said, this cuts down on wordiness and it proves to your professor that you're actually gaining some scientific vocabulary just from hearing them lecture because probably they're using these terms in their lecture. So hearing you repeat those back are really going to show your knowledge of the experiment and everything that you're dealing with. Another useful term are the words homogeneous versus heterogeneous, which are pertaining to mixtures. To say a mixture is homogeneous means that it would appear to be all one thing. Like chocolate milk would be a homogeneous mixture. We know that there's more than one thing in that cup, but it appears to be all one substance. That would be considered homogeneous. Versus heterogeneous would be a mixture more like water and marbles in a cup. We can clearly see the different parts of the mixture present in our cup. And lastly, properly identifying the tools and glassware and et cetera that we're using in the lab helps to make your lab report more concise. Although you could say that we aliquoted this liquid into a tube, that tube could be anything. And if the fellow scientist who is reading this paper wants to repeat what you have done, which is the point of our methods section, if they were to repeat what you had done, they might need to know very specific things, like what kind of tube did you use? Eppendorf tube is an example of one of these types of tubes. These are generally the small plastic tubes that you're using probably right now in Principles 2 lab, or I'm sorry, Intro to Cell and Molecular. The name change always confuses me. But so now that we've told them everything about what we've done during the experiment, now we can say, okay, what did our experiments tell us? What did we find out? So what kind of things would I find in our results section?
Charts and graphs.
Charts and graphs, absolutely. Tables and figures are a huge part of our results section. Anything else?
There's candy, people.
No? All right. Well, so our results section is where we're going to talk about our general observations and trends, where we can say, we conducted this experiment and this is what we got. Now, I'm not jumping to any interpretations or conclusions of this data. I am just simply reporting that this is what was observed, or this is the data that we collected. Any interpretation of the data should be saved for the discussion section. This is in the discussion section, which Rebecca will go over, that's where you're going to do the analysis of the data. So, like the young man in the front pointed out here, a huge part of our results section then is going to be our figures and tables, which depending on the professor and the assignment, this could be either integrated as part of the results or a separate section all to itself in the paper. When you're inserting figures and tables into your paper, first, although it's hard to have too many figures and tables, ask yourself before you insert a figure or table, does this aid in the reader's understanding of my experiment? If you don't feel that the figure or table is at all contributing to the reader's better understanding, then it might not be necessary. Second, we want to think about the organization of our figures and tables. If throughout the paper, we should try and keep to some kind of structured organization. So whether that's a sequential organization or something that just follows logically, we want to try and keep our steps of our experiment and results in the same order and keep our figures in the same order. So if in your results section you discuss proving the presence of successfully isolated DNA through a gel electrophoresis, it would, and this is the first time that you mention any data at all, it would be logical to then have figure 1, the first figure that appears in your paper, be that gel, of an image of that gel proving this data that you've mentioned. Also, a very, very important thing that you might not have run into quite yet is that all figures and tables should have a legend. So, figure and table legend. Does anybody have any idea what we should put in a figure and table legend? No? Okay, that's fine too. I'm just giving you a tip. So best way to explain this is by using an example. So this here is an image of a DNA gel that is from the Fly Lab that is so infamous amongst all biology majors. And so here we see our figure legend. It says, figure 1, gel electrophoresis results that were determined from the PCR RFLP analysis. It was determined based on the location of the bands that the single recombinant male did in fact contain both spin BBU and clue 44148. So, because we haven't read the whole paper, or you haven't even taken the class, you're not quite clear on exactly what some of these terms are, right? So, something that could be improved upon in this figure legend is making that more clear. Part of the role of a figure legend is for the reader to be able to read the figure legend, look at the figure, and be able to have that as a standalone piece of information. You should be able to look at just those 2 pieces and know why this figure is important to us. Bless you. But what we do see here in this figure legend that is good material is we've described what the figure is, what we took away from the figure, and how we obtained the figure. These are all your basic parts of a figure legend. So, say, you've got this chart in front of you and you're like, oh, I don't know what to write in the figure legend. Think back to yourself, okay, what is this chart? What is it about? First, say, this chart is showing, and tell them the information. Then maybe there was a procedure to how you obtained that information. Say, we obtained this data through, give name of procedure. And then lastly, you want to say, this data shows us that blank. Those are kind of your basic parts of a figure legend. And like I said, your reader should be able to not read any of your paper and look at just the figures and understand why you put that figure in the paper. Something good that we see here in this figure is that we see a lot of labeling going on. These are just simple text boxes put over top of the image, and these are extremely important. anytime you have a figure that is containing multiple parts to it, or it's not really clear, as especially with DNA gels, you have to label what is in each of these rows. You should always try and make your figure as clear and concise as possible through as much labeling as you need. So, which, speaking of labeling, brings me to my next category, is multi-panel figures. This isn't something you're probably going to approach and this year at your level, but this is something that will probably come up in your future, where it's appropriate to use a multi-panel figure because multiple images are addressing the same subject matter. That's really the key there. When you need multiple images to explain one concept, is a good time to use a multi-panel figure. So this is a multi-panel figure I did. I will leave out my anonymity and say I made this. So you can see what I've done here is in the figure, I have these multiple photos, but I've labeled each one as a separate panel so that I can talk about it individually in the figure legend. So in the figure legend, I say microtubules in nocadazole treated cells were stained for regrowth observation using indirect immunofluorescence. Time points were taken at 0, 10, and 30 minutes and were scored on a scale of 1 to 5 by sampling 200 random cells and averaging these scores. And then I quote image A, microtubule regrowth score of 5. All cells contain well-defined, fully formed filaments. B, visual examples of microtubule regrowth score of 4. Some cells contain excess of free tubulin, but most exhibit fully formed filaments. C, regrowth score of 3. Around 50% of cells contain excess of free tubulin, but some cells contain fully developed filaments. D, regrowth score of 2. Some filaments are beginning to reassemble, but high levels of free tubulin are present. And lastly, E, regrowth score of 1. No filaments are present, all tubulin exists as free tubulin. So in that figure legend, there was some vocabulary words that you may not be familiar with, because when I made this figure originally, it was for a paper that was meant to be directed at people who work in the field of cell biology. So they would have some of this prior knowledge to work with to interpret that figure legend and the paper as a whole. But what I did do is, although I grouped these photos together in one figure, I explained each photo individually, and I talked about why they relate to each other. They're all examples. In this case, they were all examples of the scoring method that I developed for my experiment. I also talked about exactly how I obtained this data, and I talked about some conditions as far as what these cells were treated with. In this case, it was a drug called nocadazole. Bless you. So now that your readers have all of this information about what you observed, now we can talk about interpretation.
Okay, so we're going to talk about the discussion. What goes in the discussion?
An explanation of the results in the table.
Yep.
Excellent.
So I should be a professional candy thrower. We're going to start in the discussion here. So your discussion is actually your interpretation of your results. So instead of simply restating what you got, you need to actually think about what it means. You don't want to introduce any new results here. You don't want to, get all wacky with it. But you want to make sure you're going through, you're seeing what you got, and how that relates back to your actual hypothesis or expected result. And so you want to compare your results, obviously, to your hypothesis. Do they support? Do they not support? If they don't support, what are some possibilities of why they don't? Do you have error in your experiment that could explain all of those things? And you have to relate the results to the problem question. So You can't just say, this was this, and it was this, but it had nothing to do with your actual first question. And that's, I think, understanding where your results have to go with your hypothesis is a big part of your lab report. So your discussion must include, you want to look at, you want to take a further look into your experiment. So you have the results, you've looked at how they relate to your hypothesis. Now you want to see, okay, where can I go from here? This isn't something where you can just take what you got and roll with it. You need to sit down and critically think about where you can go. And I mean, it could be as simple as doing more analysis, or if you had error, going back and correcting for the error. If you're into, I mean, the harder papers, you're gonna be doing a lot more critical thinking. You wanna have a conclusion paragraph that pulls everything together, your entire lab report together. So you wanna start with, you know, what was your problem, how you solved the problem, and then, you know, what your results truly meant in just a couple sentences. And should not be wordy. You want to keep it short and sweet, because at this point they're reading your lab report and they're kind of getting over it. Okay? They are. You're reading, you got all this stuff, and I know you guys are all in the intro labs, basically. They're reading a lot of papers, and at this point they're kind of like, okay. So
how do they feel, Nancy, when they get to reading the discussion?
Well, since I grade, it gets tiring. I guess that's a good point to bring up, because remember you're writing for your teacher. Some of you are in my class, and by the time we do get to the end there, you've basically given us a lot of things on how things develop, but at that point, we just want to see if you're making the connections. Are you interpreting that data correctly? Okay. You've given us the data in the data section. You've walked us through the methods. You told us the background, but now we need to make sure that you're interpreting your results. And so at that point, if you're just listing more things, are you okay with that?
Yeah.
So if you're just listing more things, then you're really wasting your time in that section when you should just be using it to, as she mentions, take a further look at your data, and you're gonna really relate all the data that you've put in your report to that hypothesis you stated at the beginning introduction section, okay? And so yes, by the time, realistically, that professor just poured through all your information, and we're looking to see if you took that further look into your data to connect the results to that hypothesis. Sorry, I mean, she kind of put me on the spot.
okay, so you can use references to support your findings. So if you find another paper that has the same results kind of thing, you can use that to say, this is what we got, this is what it means, this is supported by, and then you can cite that. But you shouldn't be bringing new information at that point. It should just be something to really nail that. I'm not going to finish that statement. I don't want to say anything about coffins today. When you're organizing this, how your results are organized, you have result 1, result 2, result 3. Your discussion should flow, talking about result 1, result 2, result 3. No one wants to be jumping around. You should be able to flow through that paper. You're in the middle of the discussion. I'm going to guess that your result is somewhere in the middle of your results section. It should be very easy to go back and forth from them. So really, if you have the results section done, you should really just go through and make that discussion from that organized list. And you really need to pull everything together. So now we have these 3 things: your acknowledgements, your literature cited, and your appendices. So, anybody have any comments on those you'd like to talk about? Okay, so your acknowledgements are optional. It's where you give a shout out to anyone who helped you. Most professors don't even care if you put this in there. I know for Intro to Ecology and Evolution Lab, I literally use it as a joke with Titus. So it's totally optional. Your literature cited, you need to make sure that all of the outside information that you have put into that paper is cited. And I like to go by the rule, if you've read it, you should just cite it anyways, because your brain will remember it. And you don't want to fall into any trap with plagiarism. Next week, Allison is going to do an entire discussion about citations, plagiarism, finding information, all that stuff. So that'd be really good to come to. Your appendices is also optional. Some people don't like to format their paper with their results right in there, right in the section. You can throw them at the end. Take the extra 10 minutes and put it in there. It's not that hard, I promise. I can do it. Okay, so this general information, we actually emailed the professors, and this is what they sent us back about their biggest pet peeves with writing. And so with their structured feedback, they want you to read the comments carefully and incorporate them into new ideas in your next papers and make sure that you're not making the same mistakes. Because so many of them complain that they tell you something and then the next thing you hand in, they have to tell you it again. And so when you're writing, look back at your old work. Make sure that when you're writing your next paper, you're not making the same mistakes. You're not going to grow as a writer unless you really look back and see where you started from and how you can improve. And if you don't understand something, ask. They get really sassy with you if you wait like weeks upon weeks and you're just like, I don't know what I did wrong here. When you get that paper back, read through those comments. If you don't understand why they marked you out for something or what they wanted you to put in that section, you need to go talk to them because they will help you. It is their job. The language, clear, concise, with proper grammar. A lot of people like to just go wordy, 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 and I will admit I did this. My first genetics lab report was 21 pages long. It was horrible. It was absolutely horrible. And so I cut it down and it ended up being like 10. So you can get very concise with your words, okay? You don't have to put every single step you've done in there. And the biggest thing was proofreading. You can have a great lab report, but if an instructor is reading through it and there are grammar mistakes or it's just not set up properly and it takes them time to read, they're going to get aggravated. So you proofread it, you have the person sitting next to you proofread it, you find Megan and you have her proofread it.
I cannot proofread for you. I cannot do grammar and proofreading, but I have a very important proofreading story. So my freshman year when I first started college and I was in Dr. Titus's lab, I didn't proofread anything until one day I handed in a lab report to Dr. Titus, not knowing that a particular friend of mine had put inappropriate words into the middle of all of my sentences throughout the entire lab report. To which I then got the lab report back from Dr. Titus and with a little note saying, I don't think you meant to put this here. And I have always proofread ever since. So.
Do you have Tourette's?
Are there English teachers who can proofread?
No. We are allowed to read for content, but we cannot proofread for grammar.
So would you recommend finding a friend?
Yes.
While they're sitting next to someone, make a friend, they're your proofreading buddies. I will be honest with you, I make my roommate is a criminal justice major. She has no idea what any of my work actually means, but she proofreads them and they're phenomenal. And so they don't have to understand it, they just have to know English.
a good tip for me for proofreading your work is, believe it or not, it helps if you read it out loud to yourself. I sound like a complete moron when I'm rattling off my genetics lab report. And now I write for, basically as a graduate student, I'm writing for my PI. He's, very particular about writing, and I'd be very embarrassed if I didn't. And you read it out loud and you'll be surprised at how many things you catch. And it's not intentional in your head, it sounds fine. You're reading, your eyes are moving along with what you're thinking. But then you read it out loud, it forces you to slow down and really say that sentence you wrote. And you'll realize immediately that needs a comma here, or these are two clauses that I put into this ginormous one paragraph sentence.
Or your grammar is totally wrong.
Yeah, or your grammar is totally wrong. Do know that you do want to shy away from being informal, so if it sounds like a conversation with your buddy, it's probably a little too informal, so.
Also remember, Microsoft Word's not always your friend. It's, if you use the wrong word and it's still spelled right, it's not going to catch it. So always read through it. You don't want to do that, it's just a silly mistake.
I mean, as students, it seems like the only way I proofread my papers by myself, because I will keep thinking that my thoughts were right, is I go backwards. I start at the last sentence and work my way to the first. You really have to find something that works for you, because it's all about how you understand English, which is different for everyone. Okay? Questions?
Yep, so now we're opening up the floor to questions. Any specific questions, general questions, that you
Yes, wait,
have?
I got it. You're good.
Oh, you got it? Okay. Questions about anything at all? Yep.
Abstract, would you recommend like taking something from each section and putting them all together?
That's,
I wouldn't say directly, I would paraphrase maybe. I wouldn't take direct sentences and copy and paste, no.
but locating one sentence in that section that kind of summarizes the section as a whole is a good idea. But yes, make sure you paraphrase. Professors will find it very obvious if you've just taken the same sentence from multiple sections and put it in different places.
Make sure it's a new sentence.
Any other questions?
Any lab reports you're doing that you need help with?
Well, I have a couple questions. I'm gonna quiz you guys a little bit, because I think there's some main takeaways that I think these, granted, is not a fabulous presentation. And they brought up very good points, and I kind of want to start off with the abstract. So you brought up bringing a sentence up from each section. But what were the three things for sure that you should include in an abstract? Who can tell me? Three things. You're about to say it, go.
So like, what's your data, I guess? Just
Summary on the subject.
Okay.
Understand that you're like gonna be researching or like a lot of things to know.
Okay, Miles, the three questions.
What did you do? How did you do it? What does it mean?
Okay, that's it. If you can distill that abstract to three things, you can really get the point across with very little words.
I mean, I've seen abstracts that are literally like 4 sentences.
Yeah, it doesn't have to be long, you just have to address those three questions. So,
You know, just on that note, I always like to think about the abstract as the teaser for the movie.
Yeah.
Right, when you see the movie, preview and it makes you wanna go see the movie, that's a good teaser. Right, if you read the abstract and it makes you wanna read the whole paper, that's a good abstract.
functionally, the way, well, we all do research, when we're going through all these people's writing, these papers, when we're actively doing research, we're using that abstract to see if that paper contains information that we can use, okay? So if you also think about it functionally, it's gonna tell you enough information about what's in that paper, enough so that we can decide whether we're gonna use it or not. So, any question on that? Okay, introduction. Can you use a figure in the introduction?
Yes.
Yes.
You
Sure? I heard another yes. You don't mind?
I'm gonna steal all of your candy. Like, come on, guys.
Sorry. Sean got it. There you go. Yes, you can use a figure legend, but I'm gonna challenge you and think a little further here. So if you, excuse me, not a figure, you can use a figure in your introduction. Now, does that figure in the introduction require a figure legend? Who said what? I heard something. She's not allowed, she's the one on the e-board, she's our secretary. Sean?
Yes, because we need to know what data you're looking
Yeah, exactly, and does it have to be data?
No.
Does the figure legend in the introduction have to be data?
Should it be data?
Should it be data? Good point. Should that information in the introduction, should it be data? No. Yeah, we need a card below the hand. Get over here. Yeah,
So
question?
when you cite that, do you like put that in your works cited on the rubric?
Yeah, well,
You can,
Yes, yes, and come next week and I'll tell you all about it.
yes. So, and she will really get into the nitty gritty of that. Just note that you have to cite it. The numbering becomes a style, a citation style. But yes, you do want to cite it and you want to list it in that reference, okay? Now, compared to the apps,
I actually have a question about
Yeah.
that. If you put like figures or pictures in the introduction, do you still put that in the same section with the rest of the figures?
Depends on your professor. Some of them want a separate appendix that is a figure section.
Mhm.
Others want you to integrate it right into the paper. Others don't care and they'll just tell you to do whatever easiest for you.
Yeah.
So
usually you'll get a rubric or some sort of, handout before the round of do that tells you how they want it.
and these are all questions, do know they would rather you ask than them reading something that's not what they want. These are all acceptable questions, they're never too rudimentary because they know every professor might want something different. So it's okay to ask your professor, do you want this here in this way, okay? So my next quiz question here is, compared to your abstract, which of the two sections between the abstract and the introduction have citations? The introduction, she took notes, she looked, you sure you don't want Okay. I wanna give these candies away. Yeah, the introduction, remember, Miles said you're gonna have more than enough time to cite your information in the introduction, so you're gonna keep that abstract concise. Did you have something?
no.
Alright, moving on to what's the next section?
Really?
Really? Did you go
I
there?
just wanna make sure I'm following their format.
Methods.
Okay,
Methods.
methods. What's a good quiz question for methods? So is the method section a simple regurgitation of your protocol?
Yes.
Are you allowed to like refer to the lab handout for that or?
That's a good question. You wanna ask your professor for sure, but usually, and this is speaking from experience with our professors here, oftentimes they say, if there's a deviation from your manual, you wanna list it, say, this is from the manual, this is the step that's changed, and they don't want you to relist everything. But see, and you brought me to the point of that quiz question, is that you wanna shy away from the listing in general. Because what the methods is gonna do is put everything in order. We're all dealing with isolation of DNA of pseudomonas. Well, not all of us, but a lot of us here. And so remember, you have to walk us logically through the steps of each week. Okay, remember, it's a 5-week project, but it's all one experiment. So logically, they're all connected. And that's what the method should do, is connect all those individual protocols into something with a narrative so that the reader knows what each step meant and how it helps you get to your result.
More about that, to answer your question, you can cite your landing.
Yeah.
You would never copy and paste it. But you can, if they want you to put that information in, you can rewrite it, basically, simply, and then you cite it.
I've read papers just for my research that if you're using a technique it's very conserved throughout different papers. A lot of the times they'll say, we used, X technique as used by such and such, I mean, and then you'd have to go back and find their paper in the works cited to look for that technique, so.
But generally, you're gonna either write it out or skip it altogether.
Any questions on, where were we? Methods. Okay, good. So methods, results. Okay, what's a good question for results? I'm gonna stump you guys with this
How often do you need labels?
one. Ooh, that's a good question. In your results section, how often do you need labels? This wasn't explicitly said, but you can kind of use this.
Whenever you have something that's like multi-parts, or like we have like a picture that is like 4 or 5, or like a geometric basis where it makes like multiple different samples analyzed.
Do you feel happy with that answer?
I feel like that's definitely a good answer. Does anybody else have any other answers?
I think he wants candy though, but.
Sure.
You can absolutely have candy for that. Well, what I would say is the correct answer is as often as you feel it's needed. I feel like it's pretty much impossible to over-label a figure because any label you add is going to add to the clarity of that figure and what it is that the reader is looking at.
She had a very good example of that gel, and in fact, although you guys didn't know, can you pull that up? That's a very good figure.
Thanks, I tried
So-
really hard.
Look at this figure here because you guys will, I think assuming everyone is in the intro to cell molecular, but a lot of you are. So notice at the top there, WT, spin BBU, and A. At the top there, that figure, you're gonna manipulate it yourself. You will put this in PowerPoint, you will put a text block, and you will slap it right on there, okay? And that's how you're gonna generate that. That takes some doing, okay? You're gonna have to actually tailor these figures yourself. And I think that's often,
It takes time.
it takes time. And do realize that all these additional figures, these little labels, she put that in for clarity. Now, you don't know any of this information, but you do know that spin BBU and clue 44148 were mentioned. And so if it's mentioned here, that's basically prompting the reader to look for them up there, okay? And that's how you're gonna use that figure legend. You're gonna prompt the reader to put their eyes exactly in the parts they need to. Notice here, there's a couple of things we're not looking at that are not mentioned, so our eyes don't go there. Okay, so this is a way of actually guiding your reader. For example, you guys share gels with other lab benches. Half that gel is not yours. You want the reader to go to your part of the experiment. Make sense?
Yeah, I just want to mention figures, as far as altering figures, organizing your figures, putting labels on your figures, for a major lab report, a big paper, personally, I spend at least an hour doing that kind of stuff because
hours.
it is
Hours.
long and
Yeah.
tedious. Do not think that your figure section is just going to fall together. You need to dedicate time to it.
In fact, your data section, and he mentions that he writes his abstract last. The first thing I do in the lab report is organize my data.
Me too. I do the
Same.
thing.
Okay? Because that's gonna give you insight as to, you're already gonna start interpreting that data, and now you need to know, now you're gonna know information that the reader's gonna need, and you're gonna know how to interpret it. Okay? So I always start by analyzing my data.
On this, I'm just gonna tell you when I made this, the copy and paste was like my
Friend.
Oh,
Yeah.
I made one text box and copied and pasted that thing all over and just edited where I needed to. And I'm gonna tell that gel would probably, I mean, it was huge.
Yeah, that gel is like,
Yeah.
so she cropped it.
Because I don't think you wanna see the
Whole class.
well, yeah, it was just weird.
Sure.
I just remember from my genetics one, I felt like, because I do work backwards, so I'll start with data and results, so you can kind of understand it. And then like when I put it in data and results, or when you put it in like a reference index section at the end, I just referred to it, so like if I talked about it in my intro, I wasn't like, I didn't like put it 80 times, I just said like, what did I say, like blah, 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 and then in parentheses, see figure 1,
Yeah.
see figure 3, because then that way they can go through and it's not like taking up.
Exactly, that's a good point, you can refer to your own figures when you're talking about them. In fact, you should.
Always refer to your figure.
Always. So, result discussion, okay.
Oh my
So the order of the discussion should be in a different order than you presented your results. No, over here. Why not?
So you want organization, you want flow.
Yeah, so there's organization in flow. You sure? Oh, you made me walk all the way over here. I don't even know him, but like now we're not okay. I'm just kidding. So yeah, the discussion, you're gonna refer to those and interpret the data that you present in the results section in the same order, because when you're making your first conclusions, okay, the first thing you should be referring to is the first set of results you presented. It just makes sense. The reader doesn't have to keep bouncing around. Okay. So with that, I conclude, unless you have something,
I just want to tell you guys, I was talking to Quintine about this when we were,
Dr. Quinton.
Dr. Quintine, when we were doing all of this, and if you take 5 minutes out of your day to go talk to him, he will tell you exactly the order you should write this in, and any tips you need. He's so willing, so definitely, if you have for lecture, lab, anything,
That
go
goes for
grab
any
him.
professor, really.
Also-
Like,
Any professor, I find him to be the easiest one for me to talk to, but find the one you like and go with it.
Don't be scared.
When I had that lab report, the second semester, I had to do my intro and I was like so excited, I just thought it was so good. And Dr. Quintine, I think he got a 60 on it, and I was like, that one good. So I was really excited about it, so I went and talked to him and I wrote my whole lab report, and I went and met with him and he like ripped it apart and then I got a 97. So like, it made such a big difference, and if I had gone and talked to him, I probably would have gotten a 60.
Go see these professors with sections prior to turning it in, because you want them to look at it before they're grading it.
But not the day before.
Yeah, not the day before, not like a week before.
Our department is full of such great, kind, knowledgeable professors that really just, they want nothing more than to help you. So whoever you're comfortable with, go talk to them and they will help you.
They seem intimidating, but
They're not.
they're not. Okay, so with that said, this information we're gonna have available to you guys if you guys need it. We're still working on how we're gonna send it to you. That's why we guys had you sign in so that we can follow up with you. There's a survey we're gonna be sending out.
Yeah, the
okay?
Wi-Fi's actually down right now. That's what I was doing before, was like putting all your little emails in. The Wi-Fi's down. So I will send that to you guys within the next 12 hours. It's a simple survey.
about this.
If you just fill it out, please, it's gonna help us make these better for you guys in the end, so.
It's gonna
Thanks,
take you really
guys. Approach any of us with TriBeta shirts, we'll
Yeah.
have them in 2 weeks or 3 weeks.
I'm either here in the library all the time, so.
And that previously